reducing the amount of infections that we saw post-surgery. He later implemented a routine where doctors would wash their hands between surgeries, where instruments would be cleaned and sterilized before use in surgery. It took a while. You know, Lister proposed these ideas and used them in his hospitals, but they didn't catch on right away. It wasn't common necessarily for doctors to begin washing their hands. It took some time before his ideas caught on. But he was later knighted and had been described by British royalty as probably saving more lives due to these techniques here than were lost in all wars prior to that time. So, can you think of something that we use today that might bear his name? Listerine. Listerine, exactly. Listerine is, not saying that he necessarily invented it, but they gave it his name for sure. So, our goal then is to try to control microbial growth. We just finished talking about how we can, you know, enhance the growth or grow bacteria in a laboratory setting, but we may want to control microbial growth in our environment as well. A couple of approaches that we can take. We can be physical with the removal of bacteria, or we can use chemicals with the removal of bacteria. Washing your hands is a physical way of removing bacteria. Using soap or some other kind of cleaner like alcohol is a chemical way of moving bacteria. Using both of them, which should be a pretty good job of getting rid of bacteria on your hand. Not that they are sterile, but they would be absent of a lot of bacteria. A couple of terms that we want to familiarize ourselves with. Sterilization. Sterilization means that there are no bacteria on the surface. It is sterile, it is free from microbes. Even endospores are absent. Disinfection. This is elimination of most disease-causing microbes. So you could talk about something being disinfected, where you've treated it. It's not sterile. There's still bacteria there, but you've killed many, probably most of the disease-causing microbes. We disinfect surfaces and inanimate objects. Well, we may want to use a similar treatment on our skin. When we do that, we're using an antiseptic. So disinfectant would be something that we might be using on a solid surface, inanimate surface. Or an antiseptic then would be something we might use on our skin. Pasteurization. Based on some of the answers I got on the exam, I'm not clear that we understand pasteurization yet. What this is a heat treatment that we'll use for foods and things that we don't want to disrupt the proteins that are in them. So if you disrupt proteins of milk and juices and things like that, you're going to affect the flavor. But there's still bacteria there, and we want to get rid of the bacteria that are present in the sample. So Pasteur created this technique where if we heat the thing briefly, give it a brief heat treatment, it's going to kill most of the bacteria. And so that just extends the shelf life of the product, but it doesn't kill all the bacteria, so at some point the bacteria will grow to the point that they spoil the food. So pasteurization is just a brief heat treatment that kills most of the bacteria, or most of the microbes, but not all of them. Decontamination, I think your book uses a term called degerming. And this really is, just describes something that's been treated. So maybe something that's been disinfected, treated with a disinfectant, has been decontaminated or degermed. Sanitized. In this case, we have substantially reduced the number of bacteria that are on the surface. We would describe something as being something clean as being sanitized. Sanitized implies clean. You wouldn't expect to see dirt there. But it doesn't necessarily imply a certain bacterial level. We would expect that we've reduced the number of microbes, but we don't really know how much. And we wouldn't consider it sterile for sure. We know that there are bacteria there. We would just consider it a clean surface. Preservation. Now this is a means by which we inhibit or limit bacterial growth, usually by adding salts and sugars or removing available water, and salts and sugars will do that for us. And it applies to things that we're going to store. Things that we're going to store for long periods of time, where bacterial growth will, change and sour, we'll add things that will preserve them. What is it that they put in Twinkies that last forever? It's nitrates, right? The addition of nitrates to Twinkies allow them to essentially last forever. So when we were trying to decide on what to use to kill bacteria, kill the microbes, we have to consider where we are and the types of microbes that we might need to kill. So at your house, in your home, in your kitchen, daily life, you probably don't have to have a sterile countertop. You don't have to bleach it down. You don't have to bleach your cutting board every single day and stuff. Your level of control can be a little bit more lax because you're not generally going to be coming into contact with things that can cause disease. We have to be aware of the disease-causing microbes we might come in contact with, like Salmonella and E. coli and things like that. Take precautions to limit our exposure to those. But we don't have to be extreme with our cleaning techniques. However, if we were in a hospital setting, then things change completely, don't they? Because now you're dealing with people that are sick, so the types of microbes that you're going to be exposed to change. They're going to be different microbes than what you might have in your house. So the chemicals and the things that you need for cleaning are going to change. Consider a microbial laboratory. Now our conditions are completely different even from a hospital now. A hospital, you've got sick people you're dealing with, increasing types of microbes there. In the laboratory setting now, you might be dealing with more dangerous microbes, but the chance of them, getting out of the lab, something you'll have to address, but probably reduced to a certain extent because of the practices that you have in the lab. And then, of course, what about food prep facilities and where we have farming and agriculture and stuff? Again, we're talking about different types of microbes that you're going to encounter. And now with food, you've got to use different cleaning techniques or different treatments because you don't want to damage the food. What you might use to clean a surface in a hospital couldn't be used necessarily to clean, apples or fruit or something like that. So you have to take into account what is being cleaned, where you are, when deciding on the chemical or technique that you want to use. Considering the type of organisms that you're going to come in contact with. Also, For instance, if you are dealing with microbes that form endospores, and you know that, then you're going to have to use something that's going to kill endospores. You know, if you're canning, and you know that the Clostridium botulinum is a potential microbe that could be in your canning, you're going to have to create a temperature and a pressure that will kill those endospores. Okay? So you have to take into account the types of microbes that you might encounter when choosing a disinfectant or a cleaning agent. Now, there's this thing called the D value. That is the time that it takes to reduce a population of bacteria by tenfold, or by 1 logarithmic reduction. It's the decimal reduction time to reduce a population of bacteria by 90%. Okay? And so we can talk about agents and cleaners and stuff that have a D time of, say, maybe 2 minutes, or maybe 3 minutes, or maybe 5 minutes, or a few seconds. What this means then is that when we expose the microbes to that agent, 90% of those microbes will be killed in the first 2 minutes. The effectiveness of killing bacteria has a lot to do with the concentration of bacteria that you might have. So imagine now, using this D value here, if we had 100,000 cells and we want to reduce this count to 1 cell, how long would we have to expose the surface to our cleaning agent to do so? Well, in the first 10 minutes, we would reduce it by 90%, right? So we've gone from 100,000 to 200,000, excuse me, from 100,000 to 10,000. So that took 2 minutes. In another 2 minutes, we will have reduced it another 90% down to 1,000 cells. But we're still not down to 1 cell. In another 2 minutes, we're down to 100 cells. In another 2 minutes, we're down to 10 cells. In another minute, we're down to 1 cell. So it would take us 12 minutes of treatment with this particular chemical with a D value of 2 minutes to kill our population of cells, essentially, to bring them down to 1. Oh, why don't you just nitpick me now? I'm sorry. So it would take 10 minutes then to reduce this population if this was our D value for that particular chemical. How do they determine D value? Go ahead.
So you wouldn't have to reapply?
No, you wouldn't have to reapply. You would want to make sure that it stayed in contact with the agent, but you wouldn't have to necessarily reapply. How do they determine the D value? Well, they just basically will take they'll do what we would call an assay, where they might set up several tubes of cells, expose them to the agent, this one for 30 seconds, this one for a minute, this one for 2 minutes, 3 minutes, and so forth. And at the end of that time period, streak them onto a plate, look for the number of cell reduction that they might have. And at some point, you would see a 50% reduction, and that then, or excuse me, a 90% reduction, that then would become your D value for that particular agent. So we have physical treatment, we have chemical treatment. Let's step back and look at physical treatment specifically for a minute, and then we'll look at some specific chemicals that we also use for treating. I mentioned that you can mechanically remove bacteria simply by rubbing your hands and washing your hands, or rubbing a rag over a surface. All of those can mechanically remove bacteria, but we can also, in a physical sense, remove bacteria using heat. And this is pretty much what we do in laboratory when we flame our loop, as we're using heat to sterilize our loop to kill the bacteria. two types of heat can be used. You can use moist heat, and this is what we do with our autoclave. Our autoclave is a moist heat approach. And this is great when we're trying to sterilize media, because we don't want to evaporate the water out of the media. It'll concentrate the media, and it could affect the growth of the bacteria. Using a moist heat, like we do with an autoclave, will prevent that from happening, so we don't see our media evaporate. An autoclave, as I've mentioned before, will give us 15 pounds per square inch of pressure at 121 degrees Celsius. This then will kill endospores. Boiling will kill endospores, but it is a technique that we could use, a moist heat technique that we could use to kill bacteria. Pasteurization. I won't put it up here. Another moist heat technique to reduce the amount of bacteria. We can also have dry heat.